Everyone, I'm pleased to introduce our speaker for tonight to you. This is Dr. Matt Schrenk from East Carolina University, and he is visiting Iowa State courtesy of a program called Ridge 2000, which is funded by the National Science Foundation to do research on all the different kinds of processes that occur at mid-ocean ridges. And our speaker tonight, Matt, calls himself a geomicrobiologist. And so he blends the two worlds of geology and biology together. And he works on life that lives in the rocks underneath the seafloor. Matt is a native of the Midwest. He was born in St. Paul, Minnesota. And he attended school at the University of Wisconsin in Madison, where he got a bachelor's degree in geology. And then he went on to the University of Washington, where he got his PhD in oceanography. And he's an assistant professor at East Carolina University in Greenville, North Carolina, where he's in his second year. So let's give an Iowa State welcome to Matt.
Thank you very much, Shane. And you took away my punchline about North Carolina already, but that's all right. I want to thank the Ridge 2000 program for inviting me and giving me this opportunity to come back to the Midwest and present some of the details of my research, as well as the various entities within Iowa State that invited me to come here and give this lecture to you guys. I'm giving both a public lecture right now as well as a more departmental scientific lecture tomorrow afternoon. So I look forward to both opportunities in presenting this research to you at both levels. I'll be talking today about these slimy rocks like this that are found at the seafloor and beneath the seafloor in some of these high temperature hydrothermal vent type environments. As Jane mentioned, I'm at East Carolina University. This isn't some new state in the southern part of the U.S. that you haven't heard about. It's actually in the eastern part of North Carolina and a part of the UNC system. And so again, we're a state university in a small town, or really medium-sized town, much like Iowa State is. So again, thank you for having me here today. All right, so since this is a public lecture, hopefully that works well. Since this is a public lecture, I wanted to give you guys a little background, since I'm not entirely sure which sort of end of things you're coming from, both about the geo and the micro sides of things before we really get into talking about deep sea vent ecosystems. So basically, let's start with the microbiology here. Microbes, in their essence, are things that are too small to be seen with the naked eye. Generally, these microbial cells, such as the ones shown in this picture here, all these little rod-shaped cells, and some of them are circular, these are typically around 1 micrometer in diameter. Some are a little bigger, some are a little smaller. What this means is they're really about 1/25,000th of an inch. So they're very, very tiny. You can't possibly see these with the naked eye. We often use a microscope or other analytical tools to analyze these communities and learn more about them, both in terms of their structure and their composition. For the purposes of this talk, something that's really important is not just what these organisms look like, but also how they gain their energy and what they do to support their growth. And critical to this is the idea that microbes can harness energy from both chemical reactions and light. So in one sense, if you think back to how you get energy, you do this by eating organic rich foods and breathing oxygen. And by combining this chemical reaction, you're able to harness energy, produce energetic currency known as ATP, and use that to support eventually your growth. Microorganisms can use not only this couple of organic matter and oxygen, but all sorts of different energetic couples. So here's an example of the organic matter and oxygen going to CO2 and water. Some of the other reactions that we'll be talking about today are things like hydrogen and CO2 going to methane. This is called methanogenesis. So organisms make methane as a byproduct of their metabolism. Another one is hydrogen sulfide being oxidized with oxygen going to sulfate in water. So these are just a few of the thousands, if not tens of thousands, of different chemical reactions that microorganisms are known to use to harness energy and therefore put into promoting their growth. The third aspect of the microbiology that I want to touch upon, and maybe you already have a sense of this through the title and reading a bit of the background, is the fact that microbes, in contrast to animals and higher life forms, can thrive over a huge range of conditions. They don't just survive these circumstances, they actually grow best under extreme conditions in many circumstances. So some really well-known examples of this are like the hot springs at Yellowstone National Park, where you have organisms really flourishing in almost boiling temperature waters. On the other end of the spectrum, in sea ice or in Antarctica, in some of these very cold environments, there are also organisms that have their molecules and their chemistry and their metabolism specifically adapted to those sets of conditions. Thirdly, not only can they tolerate and grow well under a range of temperature conditions, but they also flourish from both low pH. Essentially, these organisms here grow essentially in battery acid, sulfuric acid, and you can see they have these streamers here are millions and millions of microbes that comprise these sort of streamer slime-looking system. On the high pH end, they grow up to about pH 12, so very alkaline situations. So again, they cover a wide range of conditions, and they're really adapted to performing best under those conditions. One aspect of the communities that you've already seen and that we'll be talking about more today is that these organisms don't just grow in liquid environment the whole time. They don't just grow in isolation, but they often congeal together and form these things called biofilms. So these occur in many different sorts of natural and even artificial situations where organisms grow at interfaces. So they're attached to solid materials, they're attached to each other, they're sitting at interfaces between different materials of different compositions. And this is a picture, if you remember back to the last slide, that picture I was talking about where the organisms were growing in the sulfuric acid. This is the same sort of biofilm from that setting. We were dripping it from a spatula here, and you can see how slimy and coherent this is. This is not just some sort of passive, easily broken up sort of material, but this is a very strong, resilient, almost tissue-like material, but instead of being animal tissue or something of that nature, it's really millions or billions of microbial cells making these things up. And they're glued together by polysaccharides or polymers of sugars. And within microbiology, we're just getting a sense of what some of the roles of these biofilms are in these habitats. For most of the past century and a half, we've been studying organisms growing in sort of liquid cultures, in liquid environments. And we're just beginning to appreciate some of the roles that these things play in resistance to environmental stresses, for instance. A lot of antibiotic-resistant organisms are found within biofilms because this biofilm structure can help organisms combat environmental stress. For some of them, they create their own habitats, so situations where they're happy growing best. There's also community interactions amongst the cells within the biofilms, which are another reason that organisms are known to form these things. But again, as I mentioned, we're just beginning to understand and appreciate the role that biofilms play in microbial ecosystems. All right, so that was like a mini microbiology primer there. We also need to give due time to the geology side of things. I just want to introduce you to the mid-ocean ridge setting as well at the same time. So the mid-ocean ridges are this 60,000 kilometer long seam, this is essentially a mountain range that runs through the center of all of the world's oceans. And this is the site where magma and hot rock is injected and input into the ocean crust, and new oceanic crust forms. And this has happened again and again over billions of years of Earth's history, and this is what creates new oceanic crust. So the injection of this hot magma and rock also serves a very important role in terms of the hydrothermal vent systems, because all this hot material stimulates fluid flow within the porous subsurface environment. So all of these environments where you have hot rock cooling really serve to draw in cold seawater from the deep sea, heat it up in the subsurface, and as soon as it gets to the correct buoyancy, that's the hot water shoots up back out into the sea floor. And so it's a hydrothermal convection cell that's driven within the porous rock within the subsea floor. And this is very important to the microorganisms that live within there, as we'll see in a moment. But what I want you to take home from this is that these are not stagnant systems. The essence of hydrothermal vents is that they're very dynamic systems and constantly active. And we'll see that again and again throughout the talk. Finally, another important consideration here is water remains liquid to very high temperatures. It does not boil at the bottom of the ocean at the hydrothermal vents until very high temperatures relative to that at the Earth's surface. So at the surface of the Earth, water boils at 100 degrees Celsius. At deep sea vents, water can stay liquid and not boil off and turn into a vapor phase until about 400 degrees Celsius. So we have a whole different set of physics and chemistry and therefore biology that are present within these systems. So here's an example of one of these superheated fluids that's been chemically altered in the subsea floor, and that shoots up onto the sea floor. This is what's known as the black smoker. So you have 300-degree fluids that have all sorts of minerals in them. And once this hits the cold seawater, all of these black minerals precipitate out, all these black and gray minerals precipitate out because of the contrast between the two types of fluids. And these hydrothermal chimneys, or black smoker chimneys, are conduits to the fluid flow from the subsea floor up onto the seafloor. And around these communities, there's a lot of different types of life specifically adapted to these sorts of conditions that are found at these sorts of ecosystems. Some of which you can even observe in this picture, like these clusters of tube worms and other life on the rocks, all this material in the background here. From a microbial perspective, though, we talked about these things are very, very small. So the chimney we just looked at is a couple of feet in diameter or so. So it's a pretty big habitat for an individual microorganism. So what we want to think about for the first part of this talk is the sorts of conditions that the microbes see at the scale of habitats that are important to them. So what we want to think about is looking inside the walls of these black smoker chimneys. And so on one hand, think of this as a cross-section through a chimney wall. On one hand, you have the hot fluid coming through the center of it at 300 or 350 degrees Celsius. On the other hand, you have cold seawater with lots of oxygen in it on the outside of the chimney. And within the 10 centimeters or so, across these boundaries, you have all sorts of different gradients in temperature and chemistry that promote the growth of different types of organisms. So we might expect certain groups of organisms to eat the foods that are present in seawater, and others to use the fuels and foods chemistry present in the vent fluids. And at temperatures above what we know of to support life, things like 200 degrees Celsius or 300 degrees Celsius, we don't really know what's going on at those temperatures or even if life can exist in these settings. So the important thing about this is that these are great systems over a small scale to study the feedback and relationships between the environment and the biology supported within it. And we'll get back to this diagram in a few slides as well. This all fits into the concept of, not only do we have these sort of thermal and chemical gradients within these hydrothermal chimneys, but the same sorts of gradients exist as we go deeper and deeper into the subsea floor. And so even though the deep sea is really cold right at the sea floor, it's about 2 to 4 degrees Celsius typically, as you go deeper and deeper within the Earth, the temperature heats up as well. And there's same sorts of chemicals are often available to support the growth of organisms. In this whole concept, this idea that there's a whole biosphere of organisms supported by chemical energy and living in the subsea floor is what's known as the dark energy biosphere concept. And the hydrothermal vents are a real end-member example of this because there's so much energy, so much chemical energy being expelled at very high rates through this dynamic system that you have this, not just microbes taking advantage of it, but a whole range of different animals and higher organisms living around these vents as well. If you're driving along the sea floor typically, you see sediments and not a lot of life. It's pretty barren, desert-like environment on the seafloor. There's not a lot of food that gets down from the surface of the ocean, and a lot of organic matter that hits the seafloor and supports life. And so when you're driving your submarine up to a hydrothermal vent, you see 10-foot long tube worms and clams and snails and fish and crabs and everything. It's really quite a stark contrast to the rest of the deep ocean. And so these are really vibrant examples of how much energy, how much chemical energy is really being output into these systems. One other thing we need to note as far as the introduction is the historical perspective here. We didn't really know deep sea high temperature vents existed until about 1977. That's when some of the first dives on the vents occurred near the East Pacific Rise in the Pacific Ocean. But some of the first observations that there was indeed microbial life supported by these systems were these thick mats or biofilms of microorganisms that grew around the vent area. Furthermore, when there are eruptions in the deep sea floor, when there's an injection of new rock into the sea floor and there's the expulsion of huge plumes of fluid from these systems, there's oftentimes blooms of microorganisms, an example of which is shown here. So if there's some sort of earthquake activity or volcanic input like lava flow, oftentimes that pushes out a bunch of hot energy-rich fluid, and that supports like a bloom of organisms like you might see in a pond or a lake or something of that nature. Except this is derived from chemical energy in the subsea floor. But to summarize up to this point, why do we, overall really care about this? Well, first of all, as I mentioned, the water stays liquid until very high temperatures, and so therefore, putting this together, these are the highest temperature habitats that we know of on Earth. All life that we know of requires liquid water, requires energy, and these are some of the highest temperature environments that sustain liquid water on the planet. Second, they've been operative probably very similar to the way they are today since very early in Earth's history. For about 3 billion or more years, the same sorts of hydrothermal vent systems have been operative on the planet. And so therefore, based both upon the geologic context of these systems as well as biological analyses of the organisms that live in these systems, it would seem that many of the organisms retained in these systems, many of the microorganisms, are probably similar to ones that might have existed on the early Earth. And then finally, as I mentioned already, we can use approaches and the data we derive from studying these systems, these black smoke or hydrothermal vent systems, to help us better understand the subsea floor environment as well. And the subsea floor beneath the oceans, so the oceans cover 70% of our planet. The subsea floor down to at least the temperature limit of life is actually a very large volume of habitat and has a lot of significant properties in terms of the influence upon the global biogeochemical budget. And so if we understand how life might work in this habitat, we might be able to extrapolate these sort of data a little further. Just to really highlight the advancements we made since the first studies of hydrothermal vents and the organisms that live within them, here's a plot of the year of isolation of different microbial cultures versus their optimum high temperature. So before the discovery of vents, where this red line is, in 1977 or so, the highest temperature life we knew about was about 70 degrees C, and this was from Yellowstone hot spring type environment. Ever since we've been studying the hydrothermal vents in the deep sea, this optimum high temperature has risen with time significantly. And so a lot of these organisms that grow optimally at 90 or 100 degrees C or even above the boiling temperature on the Earth's surface are all derived from deep sea hydrothermal vents, particularly from these black smoke or chimneys. So the two of the highest temperature organisms we know of, Pyrolobus fumari and Strain 121, were both derived from these black smoker type chimneys. And they're often associated with mineral surfaces, such as shown here. So the discovery of vents and the study of their microbiology has expanded significantly by 30 to 40 degrees, what we appreciate as the highest temperatures that life can exist. But basically, the overall idea is that we can use these systems, we can use these hydrothermal chimneys to study relationships between the environments, between life and its environments at extremely high temperatures. And shown here is one tool we use to conduct these analyses. This is a manned submersible called Elvin out of Woods Hole Oceanographic. Sometimes we use this to go down and collect our samples and survey these sites and so on. Oftentimes we use robotic submarines to conduct the work because they're more robust, they're less expensive, and they can stay down for longer. But we typically use a combination of these different tools to access these deep sea sites. And so here's a very nice picture. It's a composite. It's a mosaic of different high-resolution photographs that were taken of a deep-sea vent field. This is a whole cluster of different black smoker chimneys in various stages of their evolution. Some of these are dead, so this has fallen over after an earthquake or something. Others are spitting out black smoke in high abundance. Others are just sort of more diffusely venting fluids. And so there's a whole range of these things right around the mid-ocean ridge axes. And so what I had the opportunity to do as a graduate student a few years ago was participate in this cruise, in this sampling effort to go down with robotic submarines and essentially decapitate a couple of these chimneys and recover the samples and look at the microbes that were contained within them. One more note here is that this scale bar here goes up to about 14 meters. So some of these chimneys are 10 meters or so in height. So these are not tiny structures. They're large apartment complexes for the microorganisms that live within them. All right, so here's another example of just how big of chunks we were able to take off these things. This is just one chimney. This is called Finn in two parts here. It's pulled up with some Navy salvage type equipment onto the ship. These pieces here were about 1.5 meters in length, so they're really large pieces, especially in comparison to what microbes might actually encounter. And they were really huge, heavy materials as well. These are all composed of metal sulfide minerals, so it's quite dense material. They were some of the freshest samples we've ever recovered. Typically, you go down and collect a sample and drive around with it for a while before you recover it to the surface. But these were on deck within 2 hours after we cut them off the seafloor. And so they were still hot. They were still 70 or 80 degrees Celsius in the interior when we brought them on deck, even after being pulled through 2 kilometers of seawater. So we conducted integrated analyses combining geology and chemistry and biology on these samples. So this is a cross-section through the wall. Again, we have the conduit venting 300-degree fluids. We have the outside with the seawater. We took transects across these walls and compared the communities that lived inside to outside in the different gradients within the walls. So there's a whole gradient in temperature between 300 degrees and 2 degrees, and the chemistry between the hydrothermal fluids and the seawater within these settings. The other thing to note while you're looking at this is that earlier we saw a picture of tube worms on the outside of these structures. And oftentimes what happens as these sulfide structures grow is that tube worms on the outside get encased and mineralized inside the structures and essentially become fossils. And so you can see if you look very closely at this image, sort of channels created by the empty tube worm casings that have been fossilized actively within the system. And this is just an example of the mineralogical map. So we didn't just take a picture, but we mapped out where the different minerals were. This helps tell us about the conditions within the wall. Getting back to this diagram, our hypothesis, science is all about hypothesis testing. And so our hypothesis looking at this was that specific groups of organisms, whether they're methane producers or organic matter consumers, were prevalent in different regions within the chimney wall. So we applied this model to the way we analyze these structures. The first thing I did was take an awful lot of pictures of organisms forming biofilms in different regions of these chimney walls. In some places, they form like these mushroom-like colonies that have a very nice three-dimensional quality to them. This looks almost like it's just one type of organism comprising it. In other cases, they were communities that contained different shapes of organisms attached to the mineral particles. You have filaments and circles and rods and so on. You can also see it here. And then in some regions in higher temperature zones, you have these thick biofilms forming attached to the mineral surfaces. All these little dots here are individual microbial cells. And so in some of these regions, you had very thick biofilms, at least in the microbial sense, that encased the mineral surfaces. Since a lot of microbiology these days is done using genetic methods, we also looked at the DNA contained within these communities and tried to figure out who the organisms were through that approach. The important things to note here are not what all these individual groups are and to remember these names, but rather the fact that going from the cold environments to the hot environments, you see different trends in the organisms that are present there. So if you look just at the number of slices on the pie, which represent different species or groups of organisms within these environments, you see the middle region where you're having the most mixing between the cold and the high temperature fluids supports the greatest diversity of organisms. So this sort of fits with our conclusion that there's lots of our hypothesis that there's lots of different foods available to support growth in these settings. If you also look at the colors I've used to make these charts, on the exterior, anything shaded in sort of lighter colors like green or blue is associated with seawater organisms, and so you see more of those on the outside, as you might expect, where seawater plays a bigger influence. Whereas in the inside, you have more of this pink shade, which is meant to represent things related to hot spring organisms, so high temperature organisms. So all of these things can be derived by combining genetic or genomic analyses with microscopic analyses of these communities. All right, so we cut the chimney off, we looked at it, we did our best to analyze it. We still have tens of kilograms of rock sitting in our freezers that constitute a whole other series of PhD projects for other students. But that's another story altogether. Another opportunity provided by this is by chopping the chimney off and watching it grow back, we were able to observe how organisms colonize new chimney structures and what sort of organisms constitute these structures. And so, again, looking at Finn, we cut off a meter or so of the chimney and sampled it again 1 year later, and it had grown by about a meter of rock within that 1 year time period. So the rock itself precipitates and congeals and coalesces very quickly in terms of a geologic timescale. Here's another example. This is where we cut off a year earlier. It grew greater than a meter in actually 5 days. And after it formed this initial shell of a structure, it filled in. We sampled it after about 11 months of growth when it was actually recoverable, when it was solid enough that it didn't just fall apart. And what we did here is conduct comparative analyses between the two different types of chimneys. Again, we saw earlier that there was a lot of diversity where there were the two different fluids mixed, the cold and the hot fluids. In the new structures, there was a very low diversity that consisted of completely different groups. So both of these red circles consist of the clusters of sequences that we found predominantly within the young chimney. And it was the same starting material. It was just we had a known time point to go from to look at how far it had grown in that amount of time. So the interesting thing from this plot is you have two different communities inhabiting the same structure over different periods of time. And so you can really begin to analyze what the relationships are between the conditions within those structures and the organisms you see. The complication here is most of these organisms you can't study in the laboratory. We're just able to observe that they're there, but we can't manipulate them or study their growth behavior or how they respond to anything. So that's where the next tool we use comes in. And this is what's called the sulfide microbial incubator. So basically, in this case, we create drill holes in the side of these chimneys, again with a robotic submarine. These drill holes have high-resolution thermometers deployed along their length, and next to each thermometer, each of these black dots, we put little mineral chips, little polished mineral chips that we've sterilized beforehand to allow microbes to colonize these structures. And so what we have is a coordinated temperature record across the chimney wall, along with the organisms that colonize these minerals, which are similar to the minerals that you find naturally within these structures. Before I switch to the next slide, one major important outcome out of this is that the temperature gradients across the walls, and therefore the chemical gradients, because the temperature is coming from the mixture of the chemicals, is not stable. You see fluctuations in the temperature at any given point within the structure on a daily basis, and this is most likely related to tidal fluctuations in the subsurface. And so you have organisms being exposed to very hot and very cold fluids again and again. And whatever their life is, whatever their lifestyle is, has got to be able to contend with this. So in some cases, in lower temperature deployments, we saw prolific biofilms. Each one of these cells in this sort of fractal-like structure, each one of these little dots is a separate cell. In some cases, they form colonies. In other cases, they form more dispersed structures. But we have really prolific growth within some regions of this structure, really interesting patterns that we haven't even figured out how to decipher yet within just a couple-month deployment. Another interesting thing that came out of this work is that we saw evidence of biofilms even at extremely high temperatures in the high-temperature regions of these incubators. They look very different than the low-temperature biofilms. They're these clusters of DNA and other biomolecules contained by thin tubules. So you have thin tubes connecting the different regions to one another. And we also noted that these things seem to have intact membranes, and so these are at least partially functional cells. And they seem to resemble species of microorganisms that we also have in the laboratory as well. So here's an example of biofilms that were created in the laboratory using similar species to what we saw in these settings. You again see the cells connected by these very thin tubule-like structures, forming this cobweb-like network of cells. So that story is sort of fitting together. But what we don't entirely understand is how can life exist above what we know to be its upper temperature limit? Can life really exist up to and be functional up to 240 degrees C or 200 degrees C, some of these extreme temperatures that organisms are seeing within these settings? Or are we just looking at cooked microbial biomass? Are these just microorganisms on their way to being fossilized? And that's something we've tried to get at through genetic techniques, microscopic techniques, and there's still a lot of work remaining to be done to really verify that not only are these indeed cells at these temperatures, but they're actually doing something. But some of the speculation here is that biofilm formation and biofilm physiology is really essential to the survival of these organisms at high temperature. We've talked about how it can be a defense mechanism for some organisms in medical and other settings where they see lots of toxins. Perhaps there's aspects to biofilm formation in microbes that allow them to survive these extreme temperatures. So the black smokers are just one type of many different sorts of chimneys that exist in the deep sea habitat. In some cases, like on volcanic arcs in the Western Pacific, you have these elemental sulfur chimneys, these bright yellow chimneys composed entirely of sulfur. In another case, a recently discovered vent field, it was discovered in 2000 in the Mid-Atlantic Ridge, or near the Mid-Atlantic Ridge, called the Lost City. It's comprised entirely of calcium carbonate minerals. So this is like limestone, essentially. And some of these towers are up to 60 meters in height off of the seafloor. So they're some of the tallest chimneys we've ever seen in the deep sea environment. So again, here's a map of where this is. It's 1.5 kilometers off the ridge axis, so it's not right on the mid-ocean ridge, but it's slightly off the ridge axis. It's in ultramafic ocean crust, so this is really deep earth rock that's been exposed through faulting and exposure onto the seafloor, where it reacts with the water. There's a lot of this mineral olivine in it that's very unstable with regards to in the presence of water, and so this undergoes a reaction called serpentinization, which contributes to the unique conditions found at this site. So I was able to conduct comparative analyses of these systems compared to the high-temperature black smoker chimneys, which are typically acidic. These systems were very rich in volatile or gas-rich hydrothermal fluids. They contained a lot of hydrogen and a lot of methane. They had more moderate temperatures, up to 93 degrees Celsius, still pretty hot for us, but they had very high pHs, up to pH 12. So typically the high temperature events were in the range of 2 to 4 degrees, or 2 to 4 pH, whereas these were 10 or 11 or 12 in terms of the fluids they were venting. They also contained hardly any CO2, which many of these organisms used to make their biomass because it was all precipitated out in these carbonate structures. So the interesting thing here, and the really drastic comparison to the sulfide chimneys, is every single organism we encountered in this system, in different years, in different settings within these chimneys, all fell into one very narrow species group within the methanogenic archaea. So it looked like it was a pure culture. It looked like it was a clone of one type of organism forming the biofilms within these chimney systems. Upon further examination, we've seen a lot of interesting community behavior and interaction, some of the stuff I've talked about already within these biofilms. And it's a really interesting contrast in two different extreme environments that support different levels of diversity entirely. And so this is really work we're continuing to pursue in a lot more level of detail. I also want to note that not only do we have these fancy titanium incubators with thermometers all over them, but we've even been able to construct low-tech incubators from materials we find at Home Depot or Lowe's or whatever. These are like drainage pipes that we filled with sterilized rocks and watched organisms colonize in the deep sea environment. Some of the reasons you do this are because you just don't know if you're going to get it back. I mean, it is the bottom of the ocean after all, and if your submarine drops it, then you're out of luck. But a nice thing about these serpentinization systems is we also have terrestrial analogs to these sites, things called ophiolites, where old ocean crust has been pushed up onto the continents, where we can study some of the same processes with a lot more access. So one of these sites we work at is in Newfoundland, Canada, called the Tablelands. It's this environment here. It's also seeping out pH 11 fluids with very similar properties to that we see in the bottom of the ocean. Finally, we can also culture stuff from these systems. Basically, culturing is our way of taking things from the environment and getting them back in the lab so we can study them. For any of you that have ever seen a Petri plate, though, it's very easy to get colonies to grow on that because you're providing them with a very rich protein source, oxygen and air is often present, and so that's what you select for, a combination of organisms that grow really well on organic matter. But so in terms of culturing stuff from the vent environment that forms biofilms, we need to think to what is actually relevant to the communities that exist there. So some of the things we're looking into are high pressures, some of the indigenous minerals that we find at the deep sea vents, and also incorporating fluid flow. We've talked about how these systems are very dynamic. And these are all considerations that you can't really mimic on a Petri plate. So one of the things we do is we make the environments anaerobic. We take away the oxygen, and we incubate at high temperatures. And in doing so, we've been able to isolate some organisms that are biofilm formers. This is called Ax 9959, aka the snot bug. It produces these really thick clots of bacterial cells and biofilms in pretty much overnight, just on a diet of seawater, hydrogen, and CO2. We've also conducted experiments in the lab where we look at the development of biofilms over time and really pursued this idea that there's temperature fluctuations because of this mixing we've been talking about. So you have tidal fluctuations, basically cooking and cooling these biofilms. Imagine, maybe it's not that hard to imagine here, but going between a really hot environment, really cold environment again and again, you have to sort of adapt yourself to deal with those conditions. And there's some evidence we have from this work that these biofilms indeed are producing communities that can tolerate the two extreme end members. So here's an example of that. A clonal population, one group of organisms over time after being cooked and cooled again, starts to differentiate into different types of cells within the biofilm clusters. You have really tiny, poorly staining cells, and you have larger cells immediately adjacent to one another. So, to sort of summarize some of these things a little bit further and tie all of these ideas together, we've looked at high temperature chimneys in two different stages of evolution. We've looked at carbonate chimneys. We've conducted laboratory experiments, and we've even taken our experiments to the field and stuck things inside the chimneys and watched what happens. All of these things are contributing to our understanding, a better understanding of biofilms in these deep sea vent environments. Now, touching upon some of the key topics that are derived from this work that we brought about in the beginning of this lecture. First of all, what are the implications for this stuff of this work for microbial evolution? Well, if you look at this phylogenetic tree, this is comparing the evolutionary history of all different classes of organisms. You see animals, humans, chimpanzees, elephants, and whatnot are all the little tiny branches on this tree way up here in the eukarya. Right? So that in terms of relatedness, we're all pretty tightly related. All the organisms that branch off first, which haven't evolved quite as much and are more closely related to whatever our common ancestor was, all seem to be thermophilic. All of these organisms have names like pyro, thermo, methano, and so on. And so there's some clues through this sort of analysis as well that these deeply rooted organisms, these ancient organisms, prefer high temperature environments. And now that we have a sense of who's there through these studies of the deep sea vent chimneys, we can drill down through these evolutionary relationships, through gene sequences, and relate them to their functions through their biochemistry and their behaviors. With the premise that maybe life actually originated these systems. They've been around for a long time, they haven't changed much, and they retain some of the most primitive organisms that we're aware of. Biofilms are the mode of life at these vents, and so maybe a single organism didn't have to evolve its own biochemistry initially, but instead you have groups of organisms acting cooperatively at a mineral surface, conducting some of the early biochemical reactions before you had all this diversification. And some of the support for this is that actually if you look at minerals in the absence of organisms that are found around hydrothermal vents, many of them can catalyze the synthesis of organic materials from inorganic materials. These surfaces are catalytic mineral surfaces, and in both observation, theory, and experiment, all these things have been shown to be great catalysts for the formation of organic molecules. Furthermore, if you look at a biofilm, this is a microbial cell from a deep sea vent. All around it is a polymeric polysaccharide biofilm. All these little black squiggles are nanocrystalline minerals. So you not only have big, wafers or surfaces of these minerals, but they're really nanocrystals with lots and lots of surface area embedded within these polymers that can perhaps catalyze these reactions at even higher rates. So all of these favor the idea that maybe life originated within a community as opposed to an individual organism. Second, a thing that I'm really excited about, especially, is this idea that that hydrothermal vents provide us with a window to look at subsea floor environments. And the reason that subsea floor environments are so important is that if you go through and you sum up all of the organisms that exist in the various habitats around the Earth, in the ocean, in soils, in mammals and in the subsurface, the amount of cells in the subsurface environment is around 10 to the 30th. It is a huge proportion relative to any other habitat. Something like 50% of all the microbes on Earth likely exist in the subsurface beneath the oceans and the continents. A couple of slides I really like with regards to this, just to reinforce how little we know about these systems, this huge reservoir of organisms that we're just beginning to study, is looking at this map. So what this is all the ship tracks of scientific cruises that have mapped the seafloor over the past 100 years or so. So you see some places like Hawaii and Galapagos and Mid-Atlantic Ridge have lots of ship tracks, all these white lines, because those are real hotbeds for scientific analysis. But there are huge regions, especially in the southern hemisphere, where we know almost nothing about the structure of the seafloor, its topography, its shape. In fact, we know the surface of Mars better than we do our own seafloor, because Mars is not covered by kilometers of water. And so if we zoom in on this region in the South Pacific and superimpose that upon a map of North America, a map of the U.S., and compare it to the interstate highway density in the same region, it's about the same. There are hundreds of kilometers across, everywhere that we have almost no information about the sea floor in terms of its structure or what's present there. And this is let alone having any information about the subsea floor and what's going on at depth within those systems. And so studying deep sea vent chimneys really provides us with an opportunity to develop tools and approaches to deal with better exploring that subsurface environment. So, we know that organisms exist down to 4 kilometers beneath the continents, 2 kilometers beneath the seafloor. There are huge reservoirs of nutrients like nitrogen and phosphorus, as well as carbon equivalent to all plant life at the Earth's surface. But most of these organisms we haven't yet obtained in culture. Finally, another hat I wear in the science I do is to be what's known as an astrobiologist. And in essence, this is just sort of taking a multidisciplinary look at the science that we do. But when you compare this to the work we're doing at the hydrothermal systems, one thing that pops out is that if there is life elsewhere in the universe, even within our solar system, hydrothermal systems are likely a very good place to look. Some of the missions and analyses of Mars and its atmosphere and its planetary surface are focusing upon hydrothermal type systems. Even on Jupiter's moon Europa, there's evidence that there's hydrothermal activity in the oceans beneath the icy crust that forms the surface of the satellite. And furthermore, one really interesting that's coming out these days is the discovery of all sorts of new extrasolar planets. So these are planets orbiting around other stars elsewhere in our universe, many of which are rocky, much like our Earth, and provide us with many more opportunities to at least sustain hydrothermal systems. So anyway, in summary, we're armed with genomic tools now that are just allowing us to approach the frontiers in environmental microbiology and really begin to have sufficient resolution to get at what's going on in some of these habitats. The biofilms in these systems may have properties that facilitate the success of organisms at very high temperatures beyond those, beyond the known upper temperature limit of life in the laboratory. And certainly the understanding of the surface attached way of life is important to understanding the depths of the subsurface biosphere. If it is important, and if there are organisms at 2 kilometers, 4 kilometers, even 10 kilometers below the seafloor, they're likely to be attached to rocks. And so we really need to understand what is unique about that relationship and what the ecology of that relationship is. So, finishing up here, acknowledging things. I absolutely like to acknowledge the NSF Ridge 2000 program for supporting my visit, along with my hosts here at Iowa State University. I'd like to thank NASA, the NASA Astrobiology Institute, for supporting a lot of the research I do. as well as my home institution at East Carolina University. A lot of the hydrothermal work we do is in collaboration with colleagues in the Hydrothermal Vent Working Group at the University of Washington in Seattle. And also, if you're interested more in this research, potential opportunities, or news about this research we're doing, please visit my website at ECU to learn more about it. And feel free to contact me if you have any questions. Thank you very much.
Does anyone have any questions for Dr. Schrenk?
Yep.
Want to use the mic?
Is this on? Yes. I was wondering, like, with the life down on the seafloor, are you using it as an indicator about maybe how life began, sort of, but those life forms evolve so fast. I mean, is that really a good indicator of maybe the first life? Because
Which life forms evolved really fast?
the single-celled bacterial type organisms that you're studying, I mean, don't they evolve really fast and change really fast?
Well, they grow really fast, but the point of the plot is that compared to where we're at in the diversification we're seeing in the animal world, the stuff at the deep branches of the tree hasn't evolved very much in a relative sense. It's a lot closer. The length of the branches from the root to where we are is a lot greater than from the root to where the thermophiles are. But certainly we don't know how long it took to make an organism from scratch at all, and I'm not even sure how good of a handle we have on that, even after 50 years of research.
With these high temperatures that these bacteria are living at, how does that affect like their size or the rate of diffusion and that sort of thing?
Well, certainly, a lot of the transport rates are faster, and actually a lot of these organisms have specialized membranes relative to organisms that grow at more moderate temperatures, which help them maintain stability at very high temperatures. It's also seen at the low temperature end, too. So you have problems with membranes being too fluid and allowing things to be too permeable for the cells to really function properly. And so cells have developed biochemistry strategies within their membranes to really facilitate stability of the structures. In addition, a number of other things, like, for instance, if you've ever heard the polymerase chain reaction, that's where you're taking one copy of DNA and copying it millions of times in the lab. It's used in forensics and human science and all sorts of things. And you separate the DNA at about 94, 95 degrees Celsius. Well, these organisms can't live with denatured DNA, and so they have strategies to keep it stable at those very high temperatures so they can grow.
What kind of strategies?
Like proteins like histones and other proteins that help maintain structure. They have unique coiling, supercoiling, that helps maintain DNA stability at very high temperatures. They also have compatible solutes, sort of like the coolant and antifreeze you have in your car, that help to keep cells from boiling at those very high temperatures.